and separates plasmid DNA from gDNA in a bacterial sample. The first step in plasmid purification involves growing bacteria in a liquid culture. The sample bacteria we'll be working with is E. coli. E. coli will be grown in a liquid culture of LB broth placed in a shaking incubator. Only a single colony is actually necessary for this inoculation. While in the incubator, the bacteria will go through 4 phases. Lag phase, when the bacteria is initially introduced and cell growth is slow. Growth phase, where the bacteria grows exponentially. Stationary phase, where all the nutrients are consumed and cell growth slows. And death phase, when the cells die rapidly. The bacteria will be harvested at the peak of the growth phase, which should be between 16 and 25 hours, if kept at a temperature of 37 degrees Celsius with a shaking incubator at 200 to 250 RPM. But the science, it does not stop there, because we will now be purifying the plasmid DNA from a culture. Separate the bacteria from the LB broth by using a microcentrifuge to pull the bacteria to the bottom of the tube. The mass collected at the bottom of the tube is called a pellet, while the liquid remaining above the pellet is called the supernatant. We will now be lysing the bacteria in order to break open their cell walls and membranes to release the plasmids inside. Let's get down to the basics by using alkaline conditions, because it's a pun. The alkaline conditions are created by lysing the bacteria with a pH solution of 12 to 12.5. The alkaline solution can vary from sodium hydroxide to sodium dodecasulfate to SDS and even EDTA. SDS is an ionic detergent that disrupts the cell membrane and denatures proteins, while the EDTA is a chelating agent that removes magnesium ions and destabilizes the bacteria cells and inactivates harmful enzymes like DNase. Boom! The cell wall bursts, and the alkaline conditions break the hydrogen bonds and the gDNA and plasmids. But guess what? The science does not stop there. Since the plasmid DNA is in a tight supercoil, the separated strands of DNA remain intertwined, and despite not being connected, they stay somewhat together. Now we need to neutralize the alkalinity of the solutions. We will be neutralizing with a low pH of 3 to 5 molar potassium or sodium acetate at a pH of 5.5. At this stage, the DNA in both the gDNA and the plasmid begin to renature and reanneal, but because of the plasmid's unique design, they are able to reanneal back into stranded molecules faster than the still repairing gDNA. It is recommended that at this point you centrifuge, because the plasmids will now be found in the supernatant, while the gDNA can be found in the pellets. Now we actually have to purify the plasmids by choosing one of 2 options. The first option is to use alcohol precipitation. We will be using an absolute ethanol that is incubated at negative 20 degrees Celsius. Brr. Now we have to spin that mixture like a record in a microcentrifuge and then remove the supernatant. You have to wash the pellets in the 70% alcohol to remove the slats and centrifuge again. Then you let it air dry and then hydrate in either water or a tris EDTA buffer. After lysis, the supernatant that contains the plasmid DNA is added to a chromatography column. In the column, the plasmids stick to the silica under the high salt elution solution.